All right, so DNA, as we said in our question today, is sort of like, if you imagine a blueprint. So blueprint, if you're not familiar, is basically a plan that can be used to build a complete building. A full blueprint has all of the details about the structure of the building, the material it's made of, the electrical systems, the plumbing systems, all of the information to build that structure is specified in the blueprint. And if you have the blueprint, then you can construct that building. And that's sort of like what DNA is in living organisms. Okay? So it stands for deoxyribonucleic acid. And one thing about DNA is it's in all living organisms. From archaea all the way to humans and everything in all of the kingdoms, they all have the same genetic code. It's the universal genetic code. So we find the DNA in the nucleus of cells. Where exactly in the nucleus? What would you say?
Anywhere.
Not anywhere. Chromosomes. It is in the chromosomes, which are found inside of the nucleus. Chromosomes are made of protein with sort of DNA wrapped around it.
Kind of like a.
So, if we're thinking about the order in terms of size, the nucleus is the largest. In the nucleus are chromosomes. They are made of DNA. The strand of DNA has segments, which we call genes. So all the information about you is stored in this molecule. So just some sort of facts here about DNA. Okay. Every single cell, these microscopic cells, actually have about 2 meters of DNA, about maybe across your desk. That much, that's the length of the DNA. But it's all wound up very, very tiny into the chromosomes, which are in the nucleus. So in your cells, we have about 75 trillion or so cells that make up our bodies. That means in our bodies, we have enough DNA, if you were to stretch it all out, one after the other, go from the Earth to the Sun 65 times. So it's a huge length of material, but it's so small that it's basically invisible.
I have like really cool science fact.
Okay.
If you took all the elephants in the world and stacked them up like on top of each other and sent them out to space, they would all die.
All right. So DNA was discovered, where I shouldn't say discovered, the structure of the DNA molecule. The typical shape that we all are familiar with, the double helix shape, that was first described by these 2 gentlemen from England, James Watson and Francis Crick. They won a Nobel Prize. And they determined that a DNA molecule has this specific and unique shape. It's a double helix, a spiral staircase, sometimes called like a twisted ladder shape. And in this DNA molecule is a code for an organism's genes. So the DNA structure has sort of the sides of the ladder that are in blue here. They're made of sugars and phosphates. And they're all the same. Sugar, phosphate, sugar, phosphate, sugar, phosphate. But then in the rungs of the ladder, the steps of the staircase that you see here, there are 4 possible, what we call nitrogenous bases, that can be there. The names of the bases are adenine, thymine, guanine, and cytosine. We usually abbreviate them with just single letters, A, T, G, and C. And in the millions of DNA bases that make your DNA, the order of those A's, T's, G's, and C's is slightly different from the order that are in my DNA, which is slightly different from anybody else, which is probably quite a bit different from the order that's in an oak tree. So the order of bases is the code. Just like if you were to write a sentence, the order of the letters makes words, and the words make the sentences. So you notice in the two sides of this DNA molecule, two bases meet in the middle. There's a C here and a G here. And they always match up in a certain pattern. The bases are complementary to each other, is what we say. And you have to know the match. A's and T's are complementary. They go together. And C's and G's go together. So if you ever have an A on one side of the DNA molecule, the complementary base on the other side is a T, and vice versa. So to help you remember, apples in trees, cars in the garage. You need a way to help you remember that. A and T go together, C and G go together. Colin?
If you were to like take a single little DNA guy and make it big enough to the point where like you could grab it, would it be like soft?
I have no idea. I mean, you can extract DNA, and when it condenses in a liquid, you can do this with activities where you extract the DNA. It looks like a little white stringy strand if it's in a liquid. If it were not in a liquid, though, it would just sort of collapse. So if we just look at a strand of DNA, let's say on one side we had CTA, TCA, so forth, we could write in the matching complementary bases. So what would go with the C?
G. Hey, that goes.
And here? A,
G, T.
AGT,
He's gonna count.
ACG. So if we know one side, we could always fill in the other side because of this rule of complementary base pairing. So if you know about computers, the number of base pairs in a human DNA is about 3 gigabytes of space. That's how much. Imagine like on your phone or something, how many pictures you have, 3 gigabytes. That's all in this tiny little cell. So the way that the code works is the letters, A's, T's, G's, and C's are codes that give us our traits. For example, maybe CAT is a code for purple hair in this creature. Whereas a different code, TAC, would be a code for a different characteristic, yellow hair. So the letters of DNA are like letters in the alphabet. They can form words depending on the order. And then those words can form sentences. But even with just four letters, you have basically a limitless combination of patterns. And that's why just 4 letters make humans and bacteria and fungi and plants, because they have these different arrangements of those. So what do those letters actually do? Well, the code in DNA is a code to tell your body, to tell the organism how and when to put together certain proteins. A gene is really a code for a protein. So if we say you have a gene for red hair, for example, what we're really saying is that somewhere encoded in your DNA, we have a gene that determines our hair color. And the A's, T's, G's, and C's that you have tell your body how to make a pigment that's red. And so that's why you have red hair. The genes code for a protein which has some outward effect on your appearance. cells are being copied, when mitosis is happening to make new cells, all the DNA needs to be copied. So you want a copy of that cell, you need to copy all those letters in the DNA alphabet. Well, sometimes mistakes happen. And when a mistake happens in the copying of DNA, we call that a mutation. So sometimes one of those bases, maybe a G, gets substituted for a different letter. That's called a substitution. Makes sense? One letter accidentally got substituted for another. Other times, a new letter gets inserted in to the DNA. That's called an insertion mutation. What do you think we call it when a base gets removed?
Extroversion.
Not quite.
Excursion.
What's the opposite of insert?
Outside.
Oh,
Exertion.
delete.
Oh.
A deletion is when a base gets removed from the strand. So if we're changing the actual letters of the DNA code, that also means we're changing the protein that it's a code for. Okay? And that protein might not work properly anymore. There are some diseases that are caused by a single mutation. Cystic fibrosis, for example, is a lung disease. One of my friends from high school had cystic fibrosis. And it's a genetic mutation when, because of one change, the mucus that's produced naturally in our lungs is different because the protein in it is different and it's thicker, and it leads to a person being almost constantly congested, getting lung infections more frequently, and a whole host of other things. My friend passed away when she was 21 or 22 from cystic fibrosis. And so it is, again, it's caused by a mutation. One difference in that DNA can have a big impact. So to review, DNA stands for?
Deoxyribonucleic acid.
Yes. Raise your hand, please. Somebody else tell me. What's the name for the shape of a DNA molecule? Abby?
Double helix.
Double helix. Who are the two scientists that won a Nobel Prize for describing the shape? Not Lewis and Clark.
James Watson and Francis.
Watson and Crick. However, there is some there was some controversy because there was a woman that was also studying DNA. She was making X-ray crystallography images of the DNA molecule. Watson and Crick saw those images, and actually that led them to their changing their models of DNA and actually making the correct model, which they won the Nobel Prize. However, Rosalind Franklin, the scientist that took those images, did not receive the Nobel Prize, and in fact was not really credited with helping in the discovery of DNA at the time. Now most people know about Rosalind Franklin, and but yeah, she provided some of the important work that helped in the discovery. So the four bases are, what are their full names? Raise your hand if you tell me. Don't look back, don't look back. Don't look back at it. Jack?
Adrenaline, cricket, garage.
Alright, so T, A, G, and T. Thymine, adenine, guanine, and cytosine.
I got
A and T always go together, G and C always go together.
I just pronounced
Alright,
it
turn around.
wrong.
Okay. So go ahead and write out the complementary bases that would go along
No, GTA.
with this. What's that, Ashley? You want to answer?
Are we getting caught? No.
what they stand for? I'm not sure. I'll tell you
Like
tomorrow.
guacamole.
All right.
You have to-
So what is the actual strand that's complementary to this? Raise your hand if you can tell me. Ali.
GTA, GTC.
Yes. Good job.
Oh,
Interesting movie. It's old, but it's actually pretty good. It's called Gattaca.
wow.
It's about a future in which people in society are sort of segregated by whether or not they have been genetically engineered or not. We'll talk about what that means in a minute. All right. Now, scientists now can actually manipulate the genes of living organisms. So the general area in which scientists are studying this is called biotechnology. It's basically using modern technology to alter the genes of different organisms. However, it did not start in modern times. Humans have actually been changing or altering the traits of organisms for a long time. It started with what we call selective breeding, when we choose which organisms can breed. We can actually change the traits of an organism by repeating this many times. Humans have done this with dogs. All dogs descended from one species, but because of selective breeding, by humans choosing which dogs could breed together, we've created all the various breeds that we have today. All of our agricultural crops, all of our livestock, they all started as wild animals. But humans, by choosing certain organisms to reproduce, have created all of the crops and organisms, vegetables, fruits that we know today. And the way this works, if you have, let's say you're trying to grow some beans, and you have one type of bean plant that's resistant to a fungus, you have a different bean plant that also grows large pods. Well, by crossing them, you can get some offspring that hopefully might have both of those combinations. And you repeat this generation after generation, and you can breed a wide variety of organisms. These are all carrots. They have been selectively bred to be all different colors.
There's green
Chad?
carrots.
Aren't scientists doing something like that to like make like extinct species?
So we'll talk about that in a minute. So a modern way of doing this is using something called gene splicing. I don't know, if you pay attention, you might have heard in the news recently about CRISPR-Cas9 is a genetic tool that scientists could use to edit the genes of a person. So one of the hopes is that scientists could use CRISPR-Cas9. For example, my friend from high school, they are looking to try to be able to change the genes in her lungs, if she were still alive, to change the genes in her lung to fix that mutation and basically cure cystic fibrosis in her. We're not quite there yet, but in your lifetimes, when you guys go to college and have a career, this is something that's going to be on the forefront of modern medicine, editing genes within a person to try to fix problems in the genetic code.
Say like far down the line, if like the technology really advanced and it was like very like mainstream, like you
Yeah.
could just go in and change whatever you wanted. Could I like go in and they find a gene that gave me red hair and then they change it to something else? Would like my hair instantly change?
Not instantly, because the pigment gets put into those hair molecules as they're growing, but I guess it would work in that way. Hold on, so one example of this. All right, guys, listen. So the technology I was telling you about fixing within someone's body a genetic mutation, we're not there yet. But we are able to cut and paste genes from one organism to another. And one area this has been done is with using insulin. So in our bodies, in our genes, we have the gene that tells our pancreas how to make insulin. Right? Tells it what amino acids to make the insulin protein. And so in our pancreas, that gene is turned on, and we make insulin. However, some people that are diabetic, their pancreas doesn't produce insulin, so they need to give themselves injections of insulin. Back 30 or 40 years ago, that insulin came from pigs. So they would take from slaughtered pigs that were used for meat, they would take the pancreas, they'd extract insulin, and that's what they gave to diabetic people to inject in themselves. It worked in humans, but there were some problems with allergies. It didn't work great. So one thing scientists did using genetic engineering is they found in humans the gene, the letters, that tell our body how to make insulin. And they cut them out. They then took a bacteria and they inserted the human insulin gene into bacteria. And now those bacteria have the gene to produce insulin, and they produce insulin. And they grow those bacteria in these huge vats. They extract the insulin, but because it came from the human gene, it's identical to human insulin. Even though it's bacteria that are making that insulin, it's genetically identical to human insulin, and that's where all the insulin people take today comes from this process. Josh?
So you said you could like change the genes. Say like in the future, like you had a kid, like you had a child, and he had autism or something like that.
So no, because these would be things you could fix, would typically, at least in the near term, things that are caused by like one or two letters that are mixed. And we still don't know the cause of autism. If we were to find it as a specific gene and we could then fix it, it's possible. But like a lot of diseases are not strictly one single gene. There's multiple genes at work. There's environmental influences and other things. So those kind of things would be more difficult to treat. But there's a lot of diseases that are caused by one single gene mutation.
Could you fix Down syndrome?
Not in a whole, because in a person with Down syndrome, every single cell in their whole body has that extra chromosome. The only way you could do that is if you did it in the embryo stage. So if you found, we're sort of getting beyond what I wanted, but so if people have in vitro fertilization, sometimes a couple is not able to conceive a child through normal means, so they might have in vitro fertilization. They take an egg cell from the mother, they take a sperm cell from the father, they fertilize it in a petri dish, they allow it to start to grow into a small embryo. They can take that and then implant it back in the woman's uterus, and she's pregnant, and she gives birth to the baby. Now, in that process, if they fertilize the egg and they look at it and say that it has an extra 21st chromosome, that would grow into an embryo and a fetus with Down syndrome. If they could remove that chromosome in the early stages and fix it then they could sort of prevent that. Sometimes in vitro fertilization, they can look at all, because usually they produce several embryos. They can look at the sex of each embryo, and the parents can decide which sex they want to implant. In the movie I was telling you about, Gattaca, they don't just screen for sex, but they screen the embryos for all sorts of different traits and illnesses and intelligence and all of these things. And so the debate in the film is about, well, should we be able to choose all of the characteristics of our child? You know, there's a question, is, okay, most people would say, well, if we could use genetic technology to fix an illness, we should do that. But then you get into the question, well, what is fixing an illness and what is sort of improving upon?
Who's creating a super?
So like if you had a child that's going to grow to who's going to have dwarfism, should you correct the gene to allow them to grow to normal height? People might say yes. Well, what if they're just going to be like very short? Then should you correct it? Or what if they're going to be average? Should you be able to correct it to make them taller than average? Should we be able to select for intelligence, or if we knew those things? So there's a lot of questions, ethical questions involved in this. Then we get into the question, well, only rich people would be able to afford this process of selective or of genetically engineering embryos. So then do we start to create two classes in society, those who can afford to have perfect children with all the proper traits and those that cannot? And what does that mean for our society? So there's lots of interesting philosophical questions you could think about when you think about this. So gene splicing has been done in lots of situations. For example, animals that glow in the dark have been created. Goats that make silk, spider silk in their milk that they then can harvest. Potatoes, which produce vaccines in their tissue. Goats that produce medicine in their milk. Corn that produces pesticides in its leaves so that insects don't feed upon it. Bananas that have vaccine in them. So there's lots of instances of ways that genetic engineering can be used. Hold on, we're not done. Why you got late for a minute? So you probably have heard of on TV, like CSI shows, DNA fingerprinting.
Yeah.
So it really has nothing to do with fingerprints. But DNA fingerprinting uses this process called electrophoresis to create a pattern. And it's based on DNA. And so if you have DNA from, say, a crime scene, and you have DNA from several suspects, if you compare the suspects to the crime scene and they match, then there's a likelihood that person's DNA was at the crime scene. And so this is also used to test for paternity. Okay? If you're not sure who the father of a child is, by doing this DNA profiling, you could see how it matches, and you could figure out who the actual father is. This technology has been used to free people from prison, people that were on death row. There was a gentleman he's from Marcy, New York, stuck the road a bit. He was in prison for, I think, 25 years for the rape and murder of a teenager. And at the time when he was convicted there, they didn't have DNA technology. But there's a project, it's called the Innocence Project. They go back and they look at cases which they still have evidence, and they try to confirm, is this person, does their DNA match that which was in evidence? What they found is his DNA was not the DNA found on the victim, and he was exonerated and was released from prison after spending 25 years. And there's lots of instances of this. What's that? Barnes.
It is so awful how like
The girl that was murdered, I think her name was Kimberly Simon. His name is Steve Barnes. If you look up and see the news articles, he was released about 5 or 6 years ago from prison. All right, so remind me tomorrow to talk. That's where I was afraid. Remind me tomorrow to talk about cloning.
Can I find the actual?
No, they have not found the actual murderer.
What if it was? What if it was him?